Hello, dear viewers. Welcome back again in Glittering Biology with me, your bio guru Alu. Here, we were discussing in the previous lecture about the sexual reproduction in flowering plants, and in that lecture, we selected about the post fertilization structures and events of flowering plants. And in the previous lecture, we just selected the development of the endosperm along with its different types present found in the flowering plants. Now, today's topic, we are just going to discuss about the embryo and its development. If we will just talk about the embryo, it is a most important part for the which is just appear after the fertilization. There are the two structures develops after the fertilization, means after double fertilization, each ovules develops into the seed, and seeds is therefore called as a ripened ovule, which contains an embryo, and that embryo develops from the zygote. So it is diploid in nature. Embryo is developed at the micropylar end where the zygote is situated. Here, the development of the zygote into the embryo takes place only after the certain amount of the endosperm is formed. Why? This is providing assured nutrition to the developing embryo. The early stage of the embryo development, means the embryogeny, are similar in both monocot and the dicot plants, but their seed structures differ greatly. The zygote gives rise the proembryo and subsequently to the globular, heart-shaped and mature embryo. This structure you can just identify in the different segments of this slide. This is the representation, this pics of the developing embryo. How, what are the events are taking place under this stage? One by one we will just discuss about them, and you need to find out the that stage out of these segments of pictures. If you are unable to find out that structures here, then I refer to you, just go through with your book where you can get the clear picture of that structure. So let's start. Zygote divides by a transfer division forming two cells, that is the basal cell and the terminal cell, in which the basal cell divides by the transverse division to form many cells arranged in row. This structure is called as the suspensor. This suspensor push the embryo deep into the endosperm so as to absorb maximum food. You can just see here in the second segment of the picture, terminal cell and the basal cell is given, where in the third segment of the picture, the basal cell is showing the transfer division to form the many cells arranged in row, and that is just indicating that is just indicated by the suspensor. And I told you what is the function of suspensor there. The lowermost cell of the row is differentiated as hypophysis, which gives rise to the radicle. Means what structure I am just going to talk about, that hypophysis, that is the lowermost cell of these structure. The terminal cell divides by two vertical and one transverse division to form 8 cells. These are arranged in two tiers of 4 each. Two tiers of 4 each. You can this picture is not clear as much, where you can just see the that vertical and one transverse division for getting the two tiers of 4 each. So you can just see that structure in your book. Upper tier, just below the suspensor, what it is, that one is gives rise to the hypocotyle. The lower tier gives rise to the epicotyle, cotyledon and the plumule. Means the lower tier is the structure which one is responsible for many important part of that embryo. This entire structure is called as the proembryo, means where there is the hypocotyle, epicotyle, cotyledon and the plumule are collectively present over there. That one is called as the proembryo. Now this structure grows further and becomes heart shape. This you can just see in the sixth segment of your picture, where it is just looking little bit like the heart shape. In dicot, you know that there are the two cotyledons where while in the monocots only one cotyledon develops, and the integument of the ovule becomes hard and are called as seed coat, and the entire ovule is not now called as a seed. When seed germinates, the cotyledons are used up in providing nourishment, where the radicle is developing the root system and plumule developing the shoot system. In the basic structure, now we can just consider that what are the main parts of the basic structure of embryo. So if we will just study in details, then an embryo consists of an embryonal axis. In the middle of embryonal axis, cotyledon are present. The part of the embryonal axis above the level of cotyledon is called as the epicotyle, whereas the part below the level of cotyledon is called as the hypocotyle. And the tip of epicotyle is called as the plumule, which one development gives rise to the shoot of the plant, while the hypocotyle ends into radicle, which develops into the roots. So that is all about the embryo and its development. Whenever we will just study about the study about the monocot and dicot embryo development, then there are certain changes you will observe. So if you need to study more about the embryonal development into the monocot and dicot, then you can I will just refer to the book content. So go through with that and find out what are the changes you are noticing where there is a development of monocot embryo and the dicot embryo. Later on I would like to discuss about the fate of different floral parts after fertilization. What are the important parts we discussed in the starting of this chapter, that parts how they appear after the fertilization. So one by one I'm just going to select the part and what is the change you will observe. Will they appear as a part of that is structure or not? So first I'm just taking the sepals and petals. Sepals and petals. Sepal is the unit of calyx and the petal is the unit of corolla. Both structures fall off. Stamens also fall after the dehiscence of the anther. Style and stigma, it also fall off. Ovary, it forms as a fruit. Ovary wall, that is just considered now as a pericarp. Ovule will be converted into the seed, and funicle will be appear as the stalk of the seed. Hilum will remain as a hilum. Nucellus will convert itself into the perisperm. Outer integument will be just now called as the testa. Inner integument is called as the tegmen. Micropyle will remain as a micropyle. Synergids will be destroyed. Egg cell will convert into the embryo, and antipodal cells perish, while the secondary nucleus will become as a endosperm. So that is all over about the important structures. What is the fate of that structures after the fertilization? In next lecture, uh, you will just study about the seed in the post fertilization event. Thank you everyone for today. That much is.